Welcome back to my channel, and today in this video we are going to study about phagocytosis. Now we already learned that the epithelial barrier, that is the skin, as well as the mucosal and the glandular tissues that covers the openings of our body, these constitute the first line of defense of the innate immune system. This we already learned in the last video. Those who have not seen that video, I will provide the link at the end of this video. Go and check that once. So similarly, for the next line of defense, that means if suppose any pathogens somehow manage to pass the first line of defense and enter our body, then it will be stopped by the next line of defense, that are phagocytic cells that are found in our body. Now, the major phagocytic cells that are found in our body are macrophages, neutrophils, and dendritic cells. These are found in the tissues of our body. Monocytes are found in the blood of our body. So, if the pathogen enters via tissue, it will be attacked by macrophages, neutrophils, and dendritic cell. If it enter our body via blood, it will be attacked by the monocytes. So, these are the main phagocytic cells of our body that constitute the second line of defense. Now what happens is, whenever the bacteria enters, these phagocytic cells will form pseudopodia, and that's how it will, like this, it will enclose the bacteria and engulf it. It will enter the bacteria inside their cell. Now this is known as phagosome. After entering inside the cell, this phagosome will bound with the lysosome. Now lysosome contains digestive enzymes. We will see what are these enzymes and how they function. So these lysosome contains digestive enzymes. Now the function of digestive enzyme is to break down. So what will happen? These bacteria or the pathogens will get digested inside this lysosome, which is a combination of phagosome containing the bacteria and the lysosome. We call it as a phagolysosome. So phagolysosome means it contains bacteria and digestive enzyme. So what will happen? The digestive enzymes will attack the bacteria, and that's how the bacteria will get broken down into pieces, and this is the soluble debris which get exocytosis. So that's how any phagocytic cell kills any enteric pathogens. Now the question is, how phagocytes recognize microbes? This also we learned already, that in the microbes they contain some specific patterns on their cells, and these patterns are recognized by the cells of our immune system. Now because these patterns are present on the pathogen, we call it as a PAMP, that is pathogen associated molecular patterns. Or because pathogens means these are microorganisms only, so we can also say them by the name MAMPs, that is microbe associated molecular patterns. And the cells of our body contain receptors, and these receptors recognize these patterns. So these receptors are known as PRR, that is pattern recognition receptors. So in our cells we have pattern recognition receptors. These receptors are specific for the patterns that are present on the pathogens. So these patterns that are present on the pathogens, we call it as a pathogen associated or microbe associated molecular patterns. So that's how our cell identify or recognize the microbes. Now this is the table which is showing what kind of receptors are present in our body and what are the patterns by which they recognize the pathogen. So this is like CLR, that is C-reactive lectin receptors, or we can say C-lectin receptors. Now the example of such receptors are mannose receptor. So mannose receptor, that means what it will recognize? The mannose. Mannose are found on the surface of bacteria, fungi, or different parasites. So this table is showing the receptors of our body. Under that, what are the different members of those receptors and what they recognize. For example, under C-lectin receptors only, we also have dectin. Dectin means, from the name only, you can say dextran type, anything related to glucose type. So that means it will recognize the pattern, that is B-glucans. B-glucans are specific to fungi and some bacteria. So that's how our cell will recognize the fungus or such bacteria. So this is the table that shows the patterns present on the molecules or the pathogens and the receptors that recognizes such patterns. Now this is the diagram showing the same thing only in the diagrammatic way. So this is one pathogen Now you can see here on its surface some patterns are there which is known as PAMs. Now these PAMs will get recognized by the cells of our body. Now it can recognize by any phagocytic cell, that's how it will phagocyte. It can recognize by some like this mannose binding lectin or some C-reactive protein or complement proteins. So that's how it will, what it will do, it will come and bind with this microbe because they recognize such patterns. And because they come and bound with this molecule. Now this combination, the combination of the pathogen with such molecules is then recognized by any phagocytic cell. So sometimes phagocytic cell can directly recognize the pathogens because of these patterns present. Sometimes the phagocytic cell will recognize the pathogens when such patterns are already attacked by molecules like CRP or complement proteins or MBL. So this is known as opsonized pathogen. Opsonized pathogen means because of attached with such molecules, now the process of phagocytic will be more enhanced compared to the when phagocyte recognize pathogens directly. So such phagocytic cells will phagocyte the pathogens in an enhanced way. So the killing will be fast here compared to, you can take it as a slow killing comparatively. Okay. Sometimes some antimicrobial peptides recognize the pathogens because of such patterns and they kill the cell directly. So this is the method by which our body recognizes the foreign molecule or the non-self cells or the pathogens or the germs and protect ourselves. Now how phagocytosed microbes are killed? We already learned that these are killed by the enzymes present in the lysozyme, which includes defensins and cathelicidins. The pH present in the lysozyme is low pH, that means it is acidic in nature. Acid activated hydrolytic enzymes are found in the lysosomes. For example, proteases and some specialized molecules that mediate oxidative attack, and that's how the microbes are killed. Now, oxidative attack means in the lysosomes, some reactive oxygen species are found and some reactive nitrogen species are found. As I guess you might know that reactive oxygen species or nitrogen species, these are very harmful for the cells. They damage any cells of our body. They have the capability to damage all the cells of our body. So that means they are harmful for the microbes also because microbes are also cellular in nature. So under reactive oxygen species, we have superoxide anion, hydroxyl radical, hydrogen peroxide, hypochlorous acid. So what happens is due to excessive oxygen, NADPH phagosome oxidase gets activated and converts the oxygen into superoxide anion. Now this superoxide anion in the presence of superoxide dismutase will convert into H2O2, that is hydrogen peroxide. Now from this hydrogen peroxide it can lead to hydroxyl or OH radicals, and that's how in the presence of myeloperoxidase, this hydrogen peroxide converts to hypochlorous acid. Now, similar to reactive nitrogen species also, under the presence of different other enzymes in the lysozymes, they convert L-arginine into nitrogen dioxide, that is NO2. Now, this oxidative attack on the phagocytic microbes occur inside the lysozyme, and because of this nitrogen species, the cell gets damaged. Now, where the lysozyme gets this extra oxygen, the oxygen is consumed by phagocytes to support ROS, production by NADPH oxidase, and it is provided by the metabolic process which is known as respiratory burst, during which oxygen uptake by the cell increases. These extra oxygen molecules in the lysozyme or in a phagocytic cell, it happened due to respiratory burst, a specific process, phenomenon, respiration, which is called it as a respiratory burst, is only occur in the phagocytic cell, and because of which they have high oxygen concentration, which leads to the hypochlorous acid formation. Now the generation of RNS, that is reactive nitrogen species, it requires transcriptional activation of some genes for the enzyme inducible nitric oxide synthase. That means for the production of this enzyme, they require the transcriptional activation of certain genes. So because of reactive oxygen species and the nitrogen species in the lysozyme, the cells or the microbes are killed. Now these phagocytic cells, they not only help to protect ourselves from the pathogen by engulfing the pathogen, they also helps to clear our body from the dead cells. So how they recognize dead cells? They have damage associated molecular patterns. So these are the patterns that are found in self cells only, but self dead cells. So the cells that are damaged or dead, that also has to be cleared out from the body. And that clearance is done by phagocytic cell again. So how it recognizes the healthy cells of our body from the damaged cell? By the patterns on the damaged cell, which is known as damage associated molecular patterns. So because of this pattern, they'll recognize the damaged cell of our body, and that's how it will engulf and clear the damaged cell from the body. So that's all for this video. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.